Hi students, welcome to today's video. Today let us study the procedure of gram staining. So gram staining is a method that is used to classify bacterial species into two large groups, that is gram positive bacteria and gram negative bacteria. It is the first step in the identification of bacteria, and is also a valuable diagnostic tool in both clinical and research fields. So, who was the one who invented this protocol? It was invented by the Danish scientist Hans Christian Gram. He was the one who developed this technique while he was working with Karl Friedlander in the morgue of the city hospital in Berlin. And in 1884, while examining the lung tissues from patients who died because of pneumonia, Gram had discovered that certain stains were preferentially taken up and retained by bacterial cells. He did not use the counter stain in this procedure, but it was a few years later, another scientist, a German pathologist, Karl Wiechert, from Frankfurt, added a final step of staining with safranin. So, Christian Gram didn't follow the final step, that is the staining with safranin. That step was not performed by Gram. It was some years later introduced by another scientist. So, let us move on to the procedure. Here, we make a smear of bacteria and heat fixate it on the slide, on a glass slide. And then first step is, after preparing the heat fixed smear, we cover the smear with crystal violet stain. This is crystal violet stain, like this, using a filler. We cover the smear with crystal violet stain and let it be there for 1 minute. And after that, second step is we wash it out using water, and then we add iodine, that is the mordant here. Gram's iodine is the mordant here. It is a mixture of iodine and potassium iodide. And second step is addition of Gram's iodine, and we also let it there for another 1 minute. And after that we add, third step is destaining step, that is we add alcohol, which is added for decolorizing the first stain. And last step is the addition of safranin. Safranin is another stain that is added again and let it be there for another 1 minute. And then it is washed off, and now we observe the microslides under the compound microscope for viewing the bacteria. So first, when crystal violet was added, all the bacterial cells get stained and it appears purple. Then we add Gram's iodine. Then when Gram's iodine is added, the crystal violet and Gram's iodine together form a larger complex than the crystal violet or Gram's iodine alone. So this forms a larger complex, a complex of crystal violet and iodine, and that complex is insoluble in water. Third step is decolorization step. Here some bacteria, you can see some bacteria get destained and some others retain the color. The rod shaped get decolorized and the spherically shaped, the round shaped bacteria retain the stain. And when the safranin is added, the rod shaped, which was decolorized, get stained with safranin. So we can see two colors of bacteria stained with crystal violet as well as safranin. This one is stained with crystal violet and the rod shaped are stained with this one is with crystal violet and the rod shaped with safranin. So this is the overall flow chart describing the procedure. First step is fixation, then stained with crystal violet, then Gram's iodine is added, which acts as a mordant and forms a larger complex and is insoluble in water also. The fourth step is decolorization, decolorized with alcohol. Then addition of safranin. Safranin is a counter stain here. So this is the procedure, and you can see what happens to gram-positive bacteria and gram-negative bacteria during this procedure. Gram-positive bacteria will retain the crystal violet stain, that is the first stain, while gram-negative bacteria get stained with crystal violet. Then during decolorization it loses the color, and then when counter-stained it gets stained with safranin. So gram-positive bacteria, after the procedure, gram-positive bacteria will be purple colored and gram-negative bacteria will be pink colored. So that is the difference, how we identify or differentiate between these bacteria. Positive bacteria appear purple colored while gram-negative bacteria appear pink colored. And how it happens, this is a cell, this is the difference between gram-positive bacteria and gram-negative bacteria cell. Here you can see the gram-positive bacteria, it is having this green colored region is the peptidoglycan layer. I hope you all are familiar with the bacterial cell wall. This is the peptidoglycan layer. It has a thick peptidoglycan layer in gram-positive. In gram-negative you can see a thin peptidoglycan layer is present, but gram-negative bacteria has an outer layer. This outer membrane is present in gram-negative bacteria while it is absent in gram-positive bacteria. So what happens is when the crystal violet stain is added, both these bacteria, positive and negative, get stained with purple color, and the next step is Gram's iodine addition. Then I have already mentioned this Gram's iodine forms a complex with crystal violet, and they form a larger complex than the earlier. This larger complex gets trapped in the cell wall of gram-positive bacteria and gram-negative bacteria. Then next step is the addition of alcohol for decolorization. At this time what happens is the alcohol dehydrates the peptidoglycan layer. Then this thick peptidoglycan layer gets tightened and shrinks. So from this tightened peptidoglycan layer, the crystal violet and iodine complex gets trapped in it and it cannot get out of the cell wall. While here in gram-negative bacteria, they are having only a thin layer of peptidoglycan. So the complex can easily move out of it. It dissolves the outer membrane and this thin peptidoglycan layer. So here in case of gram-negative bacteria, as it is thin and it is not so tightened and shrinkened, it dissolves and the complex can escape out of the cell wall. So that is why here the crystal violet stain gets trapped in case of gram-positive, while in gram-negative it can escape. So here it becomes decolorized. The crystal violet stain is unstained. Then the next stain, counter stain, that is safranin, is added. So that safranin, in case of gram-positive bacteria, since safranin is a light colored stain, it cannot overstain the crystal violet, so it appears crystal violet colored itself. And here in case it is, as it is decolorized in the previous step during alcohol addition, it is now unstained condition. So the counter stain gets into the cell and it appears pink colored. So that is the thing behind this. Hope you all understood. Thank you.